in this video, we're going to cover chapter 13, which is Modern Understandings of Inheritance. So we're going to look at how chromosomes are inherited through meiosis and how the genes on those chromosomes can be inherited separately or together. So let's go ahead and get started. These are the objectives for 13.1. There's only 2 sections in this chapter, so it's going to be a short video. So 13.1, we're going to talk about the chromosomal theory of inheritance. We'll talk about genetic linkage. We'll explain the process of homologous recombination, which is crossing over. You probably know it as crossing over. We'll describe chromosome creation and calculate the distances between genes on a chromosome. So if you are not caught up on chromosomes, you're like, oh, I don't really know what a chromosome is, go ahead and watch this video on amoeba sisters. Okay, so chromosomes. So this is a human karyotype. So remember, humans have 23 pairs of chromosomes. They get half from their mom, half from their dad. It's not certain on which half they get from mom or dad, but they always get one from mom, one for dad for each pair. So for chromosome number 1, let's say this one's from mom, this one's from dad. Chromosome 2, they could be flip-flopped. This one's from dad, this one's from mom. So through that randomization, we get a very unique individual out of it. Now remember that each of these, all of these chromosomes, all 23, are in every cell in your body aside from your gamete cells, which are the egg and sperm. So a cell in your lung tissue, a cell in muscle tissue, a cell in your brain, in your spinal column, in your bone, it's going to have all of these chromosomes, every single cell. Now for a fruit fly, which we're going to look at in these next few slides when we talk about chromosomal theory of inheritance, we're going to look at their genetic makeup. And so they only have 4 sets of chromosomes. They also have pairs, so that's kind of neat. But this is what they look like. They have 2 tiny ones right here. They have 2 misshapen or non-identical chromosomes here, and then they have 2 that look kind of like ours. So fruit flies, Drosophila melanogaster are what we're going to look at coming up. So the chromosomal theory of inheritance was developed by Walter Sutton and Theodore Boveri. They created this theory based on their experiments with fruit flies, so Drosophila melanogaster. It states that chromosomes carry the unit of heredity. So before this time, everybody, scientists alike, didn't quite understand how we inherited traits. So through Sutton and Boveri's work, now we had a better understanding of how inheritance works. So this is the theory that they came up with. There's 4 parts. So the first part, chromosomes come in pairs, one from mom, one from dad. So that makes sense that you get a mixture from your parents. The second part, during meiosis, chromosome pairs migrate as discrete structures. So they're not going to be inherited together from mom and dad. So let's go back to our karyotype. So it's not all of chromosome 1 is inherited from mom and all of chromosomes number 2s are inherited from dad. It's a mixture, one from mom, one from dad. Okay, so chromosome sorting is random. Each parent synthesizes gametes that only contain half of their chromosome complement. And then the chromosomes combine during fertilization to reach that same chromosome number as their parents. So we cut the chromosome number in half. We go from 46 to 23 during meiosis. And then when fertilization happens, 23 plus 23 equals our 46 again, our normal number. So fertilization restores the chromosome count, which is good. That's what we want. We want an individual that has the typical count. So Boveri and Sutton showed that they had done, what am I trying to say? Boveri and Sutton had this chromosomal theory. T.H. Morgan confirmed their theory by doing their experiments, and then he kind of went an extra mile. But he showed that this white eye mutant allele was inherited with the sex chromosome. So, sex chromosomes, X and Y, for us, we're going to just call them X and Y on the fruit fly as well. They are, only the X is the coding chromosome, okay? So the Y, while it does have some genes on it, it's not very many. The X chromosome, however, it does have a lot of genes. And so the eye color gene, or the eye color absence of color gene, is located on the X chromosome. So we're going to show you how this experiment played out. So T.H. Morgan, he was the guy, again, that is trying to figure out what Boveri and Sutton were talking about through an experiment. He had a white-eyed male and a red-eyed female. So red-eyed, notice it's called the wild type. So that's the normal type. That's all that means. And then the white-eyed male is that mutant color that is pretty rare. So if he crossed these parents and got the F1 generation, well, all of the F1s, males and females, had red eyes. So he said, oh, that's interesting. So now all males are going to have white eyes. So we crossed the F1s again and got the F2 generation. And now he has females still have red eyes. Males have half red eyes and half white eyes. So he's seeing that while their one gene can be given to males and they can have red eyes, there's still going to be like a sleeper gene or a recessive gene. So if we look over here, these are our parents. There's the white-eyed male, white-eyed female, right? So if we cross them, we get all red-eyed fruit flies, male and female. So let's take a look. Like how could that have happened if the father had white eyes? Well, remember that Y gene, that Y chromosome doesn't really code for anything. It does, but not very much. The colorblind, or the white color, eye color, is located on the X chromosome. So mom is only going to give up her X's, right? She only has X's to give up. Both of her X's, in this case, had the gene for red eyes, for the wild type. Dad, however, so he had the mutant type on one of his X's, and then he had his Y chromosome, which didn't code for that. So in order to be a male, we have to have a Y chromosome and then an X chromosome. So the X is going to come from mom, either way, and the Y is going to come from dad, because that's the only way that we can get it and have a male. So do we see that? I hope so. So these females, though, they're going to have one X from mom, which has the red color, but then they also get that X from dad that has that white color. So they're going to be a heterozygote, the females, with that dominant red and that recessive white, whereas the males can only be one or the other. They can only be a dominant red or a recessive white, which didn't show up here. Okay, so if we take one of these females and one of these males from the F1s, we're going to put them down here in this Punnett square. Here's the female with the red and the white, and then here's the regular male with red and then the Y, which again doesn't code for anything. So This first one, this regular red, is going to be just a red female because it's XX. And then this male is going to have this regular red X from mom, and then the Y from dad makes him a male. So he's just going to have red eyes. Now, if we come down here and we look at that sleeper chromosome or the recessive chromosome with that white color, then we get a female who is just like mom, is a heterozygote. And then, since males are XY, they have one X, they have one Y, that X that they get from mom is the only genetic, is the only way that they're going to get that white color, right? Because there's not another X to block that white color. And then it gets the Y from dad. So hopefully that makes sense on how XYs occur. We're going to talk about it a little bit later as well. But hopefully you understand that even though this is a recessive gene, it doesn't get just pushed out of the population. There is always that case where we can get that that expression there. So let's answer this poll question. X-linked recessive traits in humans or in fruit flies are observed in what? Now notice I don't have only males. So we can have a case where we get X-linked recessive traits in females as well. So A should be the correct answer, in more males than females. Now if we had this case here, a heterozygote female and then a white-eyed male, if we crossed them, we would get one X and one X here that have that white color. And if we have two X's, that's going to be a female. So that would be a female with white eyes. So that's the case where we could get that. However, it is still rare. Okay, so genetic linkage. So T.H. Morgan's work with that fruit fly sex linkage did show that the trait for sex and the trait for eye color were inherited together. So that means that they were located on the same chromosome. So that supports the idea that chromosomes contain multiple genes. So it's not just one gene per chromosome. No, it's multiple genes. However, some of the genes on a chromosome can segregate. So if traits are really far apart on a chromosome, they can separate during meiosis and be inherited independently. So we're going to look at that here in a second. Okay, so instances of no genetic linkage. So if I have genes on different chromosomes, they're not going to be linked together. And if I have genes that are really far apart on the same chromosome, they probably won't be linked together either because recombination can happen in between where they are located. So crossing over can occur within this large space here. So let me show you how gene linkage can occur. So if we have a pair of genes on a chromosome, one chromosome, multiple genes, right? So this particular gene set is located very close together. So it's really unlikely that crossing over is going to happen between those genes, right? So they are more likely to be inherited together. Let's say red hair and freckles. You usually see red hair and freckles together on a person. So those genes are located extremely close together. Okay, so this is how crossing over can occur. So let's just refresh on that. So if we had our genes really close together on our chromosome, we can have crossing over between those genes, but again, it's really unlikely. It's more likely that they'll be inherited together like that one. Okay, so genetic linkage. So let's talk more about how genes are linked together. So if we look at, this is a genetic map for our fruit fly. We're showing aristae, the length of aristae, body color, red eye color, wing size, and then either brown or red eyes. So there's two genes for eyes. One gives them really red eyes, one gives them more brown. And then another gene can really give that brown color or just embrace the red. So again, so this is a chromosome here. And then these are these 5 locations. And these are just the 2 variations of that gene. So those are like the alleles, right? So short or long, that's the allele for that gene. So which of these genes do you think are likely to recombine? Or which of these genes are not linked? So it's probably going to be the aristay length and the red versus brown eye shade, because they're really far apart, right? So this number, it's really, really big. It's going to be really far apart. But this aristay and black body, gray body gene is close together. So those genes could be linked. But then if we look at even this body and the red or cinnabar eyes, those are even closer together. So those are very likely to be inherited together. So if they have a black body, they're probably going to have cinnabar eyes. If they have a gray body, they're probably going to have red eyes. Okay, so I hope that makes sense. Okay, so the frequency is the chance on which recombination can occur. So the lower the frequency, the lower the chance of recombination. So this B and C has a really low chance of separating. And then this A and B has a higher chance of separating. And then the A and C has an even higher chance of separating and being inherited separately. So a pair of genes with a larger recombination frequency are likely farther apart. Okay, so A and C, 18.5%, they're the largest. So they're probably going to be located on either end of a chromosome. Where if we look at B and C, those are going to be located really close together, most likely next to each other, if that's the lowest number. Okay. So let's do a practice. We have genes A, B, G, and H on one chromosome. And these are the distances right here. So what is the likely order of the genes on the chromosome? So if we're looking, this 2 probably jumps out at you. Those are going to be located really close together. So A and G are going to be next to each other. So let's look at our answer choices. A and G are next to each other, next to each other, next to each other. So we can go ahead and cross out D because it has A and G really far apart. Okay, so let's see. I like to go for the largest number next, which is 20. So H and G are going to be the furthest apart from everybody. So A and G are close together, but H and G are not far apart. H and G are very far apart here. H and G are somewhat far apart. So we're going to go ahead and say B for right now, but let's go ahead and check these other answers or our other map units. So A and B have a map unit of 10, and then H and B have a map unit of 8. So that looks good to me, so I'll say letter B for that one. Okay, so let's do one more. So recombination frequencies are set as followed. How would they be organized on a chromosome? So go for the smallest number first. So Q and T are the smallest, so they're going to be located next to each other. And then go for the largest. So R and S are going to be the furthest apart. So Q, T, R, S, we're going to say letter A, correct? This one has Q, T. Oh, it could be letter B. Okay, so let's see. It could be letter A or B. QT not next to each other. QT's next to each other, but RS, that's not correct. So let's look at A and B. So it could be RTQS or it could be RQTS. So it's just that these flip-flop. Let's see. So R and S are really far apart. Let's go for the next smallest. So Q and S should be located next to each other. So we're going to say letter A since Q and S are like next to each other. Okay, so that's 13.1. Hopefully now you can describe chromosomal theory of inheritance. You can talk about what genetic linkage is and isn't. You can talk about crossing over and how likely and unlikely crossing over is to happen. And then talk about chromosome creation during meiosis and distances on a genetic linkage map. Okay, so 13.2, we're going to talk about inherited disorders and how they partake or how they relate to chromosomes. So we'll talk about how a karyogram is created. We'll talk about what a karyogram is as well. Nondisjunction, how that happens. We'll compare disorders in aneuploidy cases, and we'll talk about how euploidy cases and disorders can occur as well. So karyotype is a snapshot of your chromosomes. So if you ever had genetic testing done, or you know somebody who has had genetic testing done, this is likely what the doctors looked at to determine if so-and-so had a genetic disorder. Likely if you're a woman over the age of 30, your OB is going to request a genetic test if you get pregnant. And the genetic test is just to see if the baby is going to have a genetic disorder, because genetic disorders increase with maternal age. So the older a woman is, the more likely she is to have a baby with a genetic disorder. So this is what the doctor would look at when the genetic test is taken. This is a snapshot of all the chromosomes, and they'll just look to see if the chromosomes are intact, for one, if they look identical, there's not pieces missing. They'll check to see if there's an extra chromosome on one of the pairs, and they'll check to see if there might be a missing chromosome on one of the pairs. I do want to point out that no, this is not how your chromosomes are going to look when they take a picture. They take a picture of many cells under the microscope, and then they kind of cut and paste them together to form a karyotype picture like this. So no, this is not what they look naturally. This is just like a cut and paste picture that they did. So this is a female. You might have seen that already. So there's 2 X chromosomes here, and this is a euploidy individual, meaning that this individual, this female, has the true number of chromosomes for a human, which is 46. If this was a male, there would be 1 X chromosome here, and then 1 little Y chromosome off to the side. Usually looks like that size there. So karyotype creation, so more about that. So this is kind of what the doctor would look at under the microscope. The chromosomes are usually frozen at the stage at certain stages of mitosis, to where that they can see the chromosome shapes individually. So they freeze it during mitosis because chromosomes are really condensed and they're really easy to see. If they froze them during prophase, or not prophase, if they froze them during interphase, it would be really hard to make out the chromosomes because they're all unraveled. But during mitosis, they're so condensed for cell division, they can look at them pretty easily and figure out what chromosomes are there. Okay, so if we take a look at this picture here, count the number of chromosomes, well, there's an extra one here. So if it's extra, or there's not the correct number, it's going to be aneuploid. So that means that it's not true ploidy, right? So it's not going to have 46. And now if there's an extra, 1 extra in a homologous pair, it's going to be trisomy. If there's 1 missing in a homologous pair, it's going to be monosomy. So in this case, this person has trisomy 21, which is Down syndrome, and they have 47 chromosomes in every cell in their body. So instead of the normal 46, they have an extra chromosome in every cell in their body. This person over here is missing a Y chromosome, so they're going to have 45 chromosomes in every cell in their body. So this person would still technically be a female because they don't have a Y chromosome and they don't have an X either. This person here is a male, so X and Y. So you can really see that how small that Y chromosome is. Okay, so how can something like trisomy 21 or monosomy on the sex chromosomes happen? So it happens through nondisjunction, and there's two places during meiosis. So during meiosis, it's going to occur. The first part, or the first location nondisjunction can occur is during meiosis 1. The spindles did not attach to homologous paired chromosomes, and so a total homologous pair gets pulled to one side. And then that results in too many chromosomes in one cell at the end of meiosis 1. And then going into meiosis 2, we're going to keep those extra pairs, and we end up with 3 chromosomes, or one more than the normal number in this case. And then the other cell on the other side has one less because the other cell got the extra. It can also just occur in meiosis 2, which instead of the sister chromatids separating like normal, it does not separate, and one chromosome gets put into a cell there. So it's going to have an extra +1. The other side of this cell is going to be missing 1, so -1. And then the other cell during meiosis 2 is going to be normal. So which is worse? Probably during meiosis 1 is going to be worse because we end up with 4 aneuploid cells versus meiosis 2, where we only end up with 2 aneuploid cells. So there's another Amoeba Sisters video that goes over mutations and how they can happen. You can watch that. So monosomy, trisomy, polyploidy, extra chromosomes don't always cause problems. When they're in animals, they typically cause problems. When they're in plants, they typically don't cause problems, and it's kind of strange. But if we think about it, so trisomy 21, we have normal of 2 chromosomes, so they're gonna make 100% of gene products. If we add a 3rd chromosome in, they're gonna make 50% more of products. So they're gonna make 150% of gene products on chromosome 21. So that's gonna result in all the issues surrounding Down syndrome. So that's how that happens. In plants, they can have an extra chromosome on every pair of chromosomes that they have, and they're going to be fine. What actually happens is that they might have more flower petals, like these daylilies here. So normally they look like this, but you can see there's like 2 extra layers of flower petals. In fruits and vegetables, they're going to have larger fruits and vegetables, a larger yield, and they'll be more robust. Whereas like these wild strawberries that grow in your backyard, they're pretty tiny, but we've kind of genetically engineered strawberries to get bigger, and so we can have more to eat. The sex chromosome nondisjunction, if we get a sex chromosome nondisjunction, so like an XXY individual, so we have 2 X's and a Y chromosome, we can actually get a male calico cat. That's how XXY is the only real way we can get it. Normally calico cats are only females, but we can have a calico male. X inactivation typically happens on sex chromosomes. So having extra sex chromosomes is really not that detrimental to an individual. Normally, if we have extra chromosomes um, on another pair, so let's say chromosome 21, we're going to have a lot of developmental issues. Whereas extra sex chromosomes, so the 23rd pair, they don't cause as many problems. And that's because of X inactivation. So during development, one X chromosome in every cell in the developing fetus's body is going to go through inactivation, so that there's only one of those X's that are active, and so only those genes on that one X, active X, is going to be expressing those genes. So let's see if I can, oh, I don't have it. I'll show you some X inactivation later on. So in agriculture, polyploidy crops tend to produce what? Larger yields should be the correct answer, letter C. So this is a strawberry, I believe. But this is how many chromosomes they can have. So this is pair 7, pair 8, pair 9. I say pair, but there's 6. So more chromosomes just makes a bigger fruit. Okay, so let's talk about common autosomal trisomy. So remember, trisomy means that there's three chromosomes where there should only be 2. Autosomes are going to be numbers 1 through 22. Okay, so all of these occur outside of the sex chromosome realm. So Down syndrome, you heard me mention before, occurs on chromosome 21, where there's an extra chromosome. So every cell in that person's body is going to have an extra 21st chromosome. It affects 1 in 700 people. Characteristic facial features: short stature, usually shorter lifespan, some degree of mental incapability. Typical heart defects are associated with trisomy 21 or Down syndrome. Edwards syndrome would be trisomy 18. So that would be an extra one here instead of on 21. Not as common as Down syndrome. Every organ is affected, and usually the baby will pass within a year of birth because the genes associated with chromosome 18 are just so important that the body is just overwhelmed with the gene products that are produced. So Patu syndrome, also the baby will die within a year of birth, also not that common. So serious eye, brain, circulatory defects. So that's going to be what's encoded here on chromosome 13. So if we have extra genes that are encoding for things that associate with the eye and the brain, that's going to cause death. So again, trisomy 21 and other genetic disorders that result in from non-disjunction are going to increase with age. So you can see where at 20, 25, it's pretty low, but once you hit 30, the OB, the obstetrician, is going to request that you get a genetic test if you become pregnant. And then it really increases with age above 40. Okay, so let's talk about the sex chromosome trisomy. So this is on the 23rd pair where the sex chromosomes are. And so, like I said earlier, X inactivation happens. So in Klinefelter syndrome, that's an XXY, we're going to have an X inactivation, but we're going to have a sterile individual, usually small testes and feminine characteristics, but still like normal intelligence. So even though the individual is sterile, they can still live a normal life, and they're going to have normal intelligence and not really a large disorder. If we look at Jacob syndrome, XYY, we're going to have 2 Ys here, usually taller than average, low muscle tone acne, below normal intelligence. So because we don't have Y inactivation, that's not a real thing. It's going to affect more than just a few characteristics. And then trisomy X, usually normal, female, healthy and fertile. So because we have that X inactivation, typically indistinguishable from other females. So if we go the other direction and we have monosomy sex disorders, so that's one X instead of a pair or an XY, it's called monosomy X or X0 or X naught. It's the only viable monosomy in humans. Do not sexually mature. They're sterile, short stature, but still normal intelligence, so still can live a normal life. And that's Turner's syndrome. All right, so let's take a look at this one. The genotype XXY corresponds to which one? So XXY corresponds to Klinefelter's syndrome. So make sure you know these syndromes here. These are the common ones. So make sure you have it written down. Okay, so that was aneuploid disorders. So if we have like an extra chromosome or a missing chromosome, now we're going to look at euploidy disorders. So where we have the normal 46 chromosomes, but there's something happening with one of the chromosomes that's not normal. So the offspring will exhibit, if a chromosome segment duplicates itself, we're going to have some kind of mental and physical abnormalities. So if we have a portion of a chromosome that is deleted, so if we take a look at this one here, this portion up top has been deleted, so it's lopped off. That means that the genes are just completely missing from that one chromosome. We get cri du cat, which is cat's cry in French. You can see the facial pictures here. Individuals usually have severe mental incapabilities, small head, unusual facial freckles, a cry that sounds like a cat, which is how it got its name. But this again comes from deletion, and it's still a euploidy disorder. Fragile X is another euploidy disorder. So in females, this can occur. It can occur in males as well. But you can see how the end of this one X chromosome looks kind of funky. Usually it's called like a hanging thread, but there's like more repeats here that occur than normal, and that causes some form of mental incapability. But again, they would have normal number of chromosomes. It's just that one of their chromosomes is abnormal. So disorders can also occur through inversion mutations. So if something happens on their chromosome where a portion of it is flipped around, that can cause that can cause an abnormality in genetics. So if we think we have like AATT here, and we flip, or here, AATT, and then we flip it, and then it's TTAA, that's not going to code the same way. So we have to stain chromosomes to see if there is an inversion, so it's kind of hard to detect, but it is a real thing. Inversions can also cause problems with the shape of your chromosomes. So normally it would look straight, but if we have an inversion, you can see the inversion here, where this big block is over here now, then the chromosomes are going to fold really strangely on themselves. So sometimes it's really easy to pick out where the abnormality is because the chromosomes are kind of chunked up. And then we can also have a reciprocal translocation where on two different chromosomes, their part of a chromosome can be lopped off and it'll attach to another chromosome where it's not supposed to be. So cancers can be caused by this, and like schizophrenia have been linked to translocations. And that's it. So that's 13.2. And that's chapter 13. So hopefully now we have a better understanding of how chromosomes are inherited, how some genes are linked, and how disorders can occur through aneuploidy or even through euploidy and some weird abnormalities that can cause a euploidy disorder. All right, so that's it. I hope you enjoyed it.